Come with me into the lab, a whole week of experiments, admin tasks, and a lot of other things. I share with you my schedule for the whole week. At the end of the video, I will show you my 7 key steps for time management, which I apply daily as a scientist in a genetics lab. This time I take you on a whole week, and I'm curious myself what's the result, because some of these experiments are crucial to my prepared publication. They should fill some gaps and complete some preliminary data that I had, so it will be very exciting. Full of experiments. Some start today and the final result I will obtain only on Friday. Today is Monday, and I will show you my to-do list, how I plan my experiments, and how I plan my time in the lab. This is my weekly plan. Monday, experiment 1 would start, which started already the week before when I streaked out the yeast strains. Before I started the experiment, I would have a bachelor thesis oral exam. Then I would inoculate overnight cultures and cast agar plates for experiment 1 to continue on Tuesday. I would have time a little bit for office reading and literature research, and then on Tuesday I can continue working with the yeast overnight cultures and doing the actual experiment, the spotting assay. Then I would plan the real-time qPCR, which you will see in a second in experiment 2. Experiment 1 will continue on Thursday by taking an image at a 48-hour time point. The last image will be taken of these agar plates at the 72-hour time point on Friday. And then I need to evaluate the data for this spotting assay. Friday will include a 3-hour online skill enhancement workshop, and the free day that I have Wednesday is when I scheduled experiment 2 to start, which began the week before by prepping the RNA for this reverse transcription and qPCR experiment on Wednesday. In between, I scheduled also my lab book update writing, and on Thursday I would then evaluate the results of the real-time qPCR. Friday afternoon would give me enough time to put all the new data together into a progress report, which is scheduled already for the next week, and I would also use the Friday afternoon for planning the experiments of the next week. Now, data evaluation. Data evaluation means a lot of Excel calculations, then transferring the data to Prism to make a graph and apply statistics. And this all helps me to figure out, is this data meaningful? You can see in the red boxes, yes, it's meaningful. This will be data that contributes to my next publication. And I'm very happy about the results. It's Friday, and today I have to take an image of my spotting assays, the 72-hour image, and then I can evaluate the data from yesterday and today and figure out what the result is. And like I said, also this assay will contribute to my next publication, so I'm pretty excited what the result will be. Before I take the images, I will join a 3-hour online course with the topic Digital Media in Teaching. I'm looking forward to implement digital media podcasting into my teaching, and I'm curious how others apply this already and about tips and tricks and exchanging with colleagues about this topic. I'm not showing you how I take the images because I showed this before, and now I show you how I evaluate the data. Each plate will have a file, and usually to save space, I will image several plates in once, as you can see here, and then each plate has to be cropped and then saved with the specific name. After that, I will put everything in an Illustrator file where I adjust all of the data so that it's nice to look at, neat and clear. And then the labeling begins, a lot of labeling. And finally, the result of this experiment is a very important result. This spotting assay let me see genetic interactions, and in this case it's a piece of negative data. So the strain that I crossed into my mutants does not rescue the viability in the presence of DNA damaging drugs. And that's fine, that's also part of the game, generating negative data. It is an important piece of data that probably goes into the supplement of my future paper. And after a little break, I come back to the desk and start the second half of my Friday afternoon, which is to collect all the data that I generated this week, all the new data, and I select those pieces that are meaningful and representative and put them into a PowerPoint presentation for the next meeting with my boss. Based on this data, we discuss then how the project continues, and we keep each other up to date with the progress of the project. I managed to get through this week by having a concise schedule where all the tasks, the lab tasks and the admin tasks, were really planned out, and no day was overfilled with the one or the other. It was a complementation of wet lab experiments and desk jobs. Also, I prioritize tasks that are on my to-do list, which are aligned with the immediate goal of my project. All other tasks are not forgotten, but they are on a list and they just stay on the back burner. You have seen me break down larger tasks into smaller tasks. I do that so that these tasks are more manageable and that I can assign the proper time slots, because for a large task it is impossible to know how long it takes, but maybe I know that casting an agar plate takes me half an hour or an hour depending on how many, and pipetting this 384 well plate took 30 minutes. I would always track my progress, meaning I have a daily to-do list and then I check off when I have completed a task. This is also very pleasing. One has the immediate feeling of success and of productivity. Number 5 is very important. I schedule admin tasks, meaning I schedule when to read a paper, when to prepare a presentation. All of these desk jobs, I try to understand how much time does it take me to do it, and then I try to schedule that in my daily work. I use calendar management and time blocking, meaning if I have a very focus relevant work like writing a paper or working on a presentation, I would block 1 hour of time where I am not allowing any interference and where I would not have any meetings. Number 7 is important to get to the point where the time management works, and that is reflecting on time management. I reflected a lot, how long do I need for XYZ tasks, how many breaks do I need, does it work for me better to work in the morning or the afternoon at the computer, or maybe it doesn't matter. So learning about one's own requirements helps extremely to go back to number 1 and create the schedule. This was a great week. Thanks for spending the week with me, and let me know in the comments what strategies you have for time management if you work in the lab or in any other job.